Cloning Plants Using Tissue Cultures and Plant Cuttings by kscience.com. This here is a rare plant. There's not many left. So how can we use a tissue culture to produce many identical clones of this rare plant? What you'll do is you'll take some of the tissue from this plant. Tissue is a collection of cells, so you'll literally take a collection of cells from this plant. The tissue is removed. It's very important that the tissue is sterilized. This is to kill any bacteria or any fungi or destroy any viruses in the tissue. You don't want those to grow in the cloned plants. So remember, the tissue is sterilized to destroy or kill pathogens. You now prepare your sterile growth medium. This here is a sterile growth medium inside a small jar. So the growth medium contains nutrients so the plant can grow. There are also growth hormones in the growth medium. This is so when the tissue is placed into the growth medium, the hormones are going to stimulate the growth of roots and shoots. After a short amount of time, the tissue culture is going to grow roots and shoots. It's going to grow roots and shoots. Once it's got roots and shoots, you can pot the plant into sterile soil. You can pot the plant into sterile soil. So if you take many tissue samples from the rare plant, you can do this on a large scale, as you can see here. You can grow hundreds, if not thousands, of this plant, and they will all be clones of one another. So they can be made quickly, it requires little space, and these plants can be grown all year round because they're tissue cultures in a growth medium. It's question time. Attempt these questions to check your understanding. This is a spider plant, and this is an image of a real spider plant. Spider plants grow very quickly and they produce many shoots. So you can cut these off. That's why it's called a plant cutting. So you literally cut a part of the plant off. So now you've got these shoots. What you're going to do is use rooting powder. The rooting powder is going to stimulate root growth on these plants. And then when you pot them, the plants will grow roots and the shoots will grow larger. You take a cutting of good parent plants. So you take a cutting of a good parent plant, and now you've got the bud. You can use rooting powder to stimulate the growth of roots, and then you can pot it, and then the plant will then grow roots and shoots. Once it's got roots and shoots, you can transfer the plant to a bigger pot, and you should then see a new spider plant being grown. So this plant is a genetic clone of the parent plant from where the cuttings came from. So the advantages of using a plant cutting as opposed to a tissue culture is that it's more simple than a tissue culture, and the plants are produced very quickly and very cheaply. It's question time. Attempt these questions to check your understanding. Press pause to practice using those key words. The answers will follow. Press pause to go through your answers and make any corrections to your mistakes. Press pause to answer the questions. The answers will follow. And if you're stuck, just re-watch the video. Press pause to go through your answers and make any corrections to your mistakes.